Hello and welcome back to WIP's Biochemistry, and in this lecture we will be handling thyroid hormones, the synthesis, the chemical nature, mechanism of action, as well as the clinical significance also. Now, what are thyroid hormones? They are derived from tyrosine, the amino acid, as well as from iodine. So in fact, they are primarily responsible for the regulation of metabolism in our body. And there are two forms, T4 and T3. T4 is, as we know, thyroxine, which is actually tetraiodothyronine, and T3 is triiodothyronine. Now, T4 is having longer half-life. At the same time, T3 is more effective, more active, around about 3 to 4 times more active than T3 in regulating metabolism. And a deficiency of iodine can lead to what is called simple goitre because it affects the production of T3 and T4. Now, in humans, the proportion of T4 to T3 in blood is 14:1. And there is always a mechanism for conversion of T4 to T3 by deiodination using an enzyme deiodinase, 5-deiodinase, which removes one of the iodines attached to T4. Now, deiodinase is a selenium-containing enzyme, which obviously shows the significance of selenium in the need for selenium in our food. And let us see the part or the role of iodine in synthesis of thyroid hormones. So these are some of the statistics regarding iodine. The total body content of iodine is 25 to 30 milligram, 80% of which is localized in thyroid gland. Obviously, the thyroid gland is the gland which is responsible for the synthesis of T3 and T4. Serum iodine level is 5 to 10 micrograms per deciliter, and the recommended daily allowance, that is the daily need of iodine in our body, is 150 to 200 micrograms. And whatever iodine we have in our body, the sole role of iodine is formation of thyroid hormones. Now, these are the sources, mainly drinking water, and we know iodized salt is being marketed for this sole purpose, and seaweeds, now generally seafoods, and some of the food items are there which are called Goitrogens, because the presence of them in the food, excessive presence of them in the food, can lead to goitre. And these substances are thiocyanate, which is present in cabbage and tapioca, and thiourea, which is present in mustard seeds. So people who are having goitre should avoid these foods. And this is the structure of thyroid gland. And let us see the structure of thyroid gland in more detail now. So this is the thyroid gland, a butterfly-shaped or shield-shaped gland in our neck. Let us see the magnified view. There is thyroid follicles. The thyroid gland is actually made of thyroid follicles with peripherally arranged follicular cells, and inside which there is follicle colloid also, and there are plenty of vascularization as represented by capillaries here in this figure. And let us see the magnified portion. This portion can be seen here. And the yellow colored, the portion here is a follicular colloid. It is here. The follicular cell is here, and the blood circulation is represented here. So blood flows through this direction. And let us see what happens now. Primarily, thyroid hormone synthesis requires iodine, which is being circulated in the form of iodide, the anion of iodine, as well as thyroglobulin, this protein which is synthesized in thyroid follicular cells. It's a glycoprotein having 5,000 amino acids in it, 660 kilodalton protein, which is synthesized in thyroid follicular cells. So the first aim is to drag iodide through thyroid follicular cells into follicle colloid, which is done like this. So the iodide is transported into thyroid follicular cells with the help of sodium iodine symport mechanism. It's actually a indirectly active mechanism, because sodium has to be taken out of thyroid follicular cells into blood, so that requires ATP. But iodide that enters follicular cells is passed through a transporter, namely pendrin, to follicle colloid, where it gets oxidized with the help of the enzyme Thyro Peroxidase. So it gets oxidized means it becomes iodine. Now, thyroid peroxidase is a critical enzyme here. It has got multifaceted role. One of them is the first role is to oxidize iodine. So iodine now reaches follicular colloid. Now let us see how thyroglobulin is synthesized and secreted. This obviously occurs in thyroid follicular cells. From nucleus, this thyroglobulin is synthesized and released through endoplasmic reticulum. It reaches or forms what is called exosomes, and by a process of exocytosis, it is released into follicular colloid where already there is oxidized or activated iodine. And this particular thyroglobulin is having nearly 115 tyrosine amino acids in it, out of which nearly 35 amino acids, tyrosines, can be iodinated. And this iodination of thyroglobulin is the next process which also is catalyzed by Thyroid Peroxidase enzyme. And iodination is like this. Tyrosine can be attached with iodine at two positions or at one position like this, at two positions to give diiodotyrosine, or at one position like this to give monoiodotyrosine. So the iodination is like this. On tyrosine there are positions 3 and 5, third position, fifth position can be iodinated, while this tyrosine is still attached to the thyroglobulin molecule. It is still attached to the thyroglobulin molecule through peptide linkage. Now, when the third position is iodinated, it gives MIT or monoiodotyrosine. Or if third and fifth position, both of them are iodinated, it gives DIT or diiodotyrosine. So this is iodination, and iodination is followed by conjugation or coupling of these iodinated tyrosine molecules, which again is catalyzed by thyroid peroxidase enzyme. So this is why thyroid peroxidase enzyme is considered as a multifaceted enzyme here. Now, coupling is done like this. If one monoiodotyrosine and one diiodotyrosine are coupled together, it gives triiodothyronine. Now this becomes T3. And if two diiodotyrosines, DITs, are coupled together or conjugated together, it gives tetraiodothyronine. It is now tyronine. So this is T4, as we know, thyroxine. Now, still this T3 and T4 are still attached to the original parent thyroglobulin molecule, and it is stored now in follicular colloidal acinar spaces, and at times of necessity it is taken to thyroid follicular cells through endocytosis. And now this is followed by the proteolysis of the original parent molecule, namely thyroglobulin molecule. It is proteolized, amino acids are recycled, and T3 and T4 are released to the blood circulation. And before release, the T4 can be a part of T4 can be deiodinated to give T3 also. This is actually done by deiodinase, 5-deiodinase enzyme, a selenium requiring enzyme. Now, this is summarized here, this process: uptake of iodine, oxidation of iodine, and both these processes are inhibited by goitrogens and enhanced or helped by TSH. Now, iodination, then conjugation and coupling, storage in acinar spaces, and utilization, and finally release, and T4 is deiodinated to give T3. And this deiodinase enzyme it is having isoenzymes, and this isoenzyme this actually can deiodinate at the terminal tyrosine to give triiodothyronine, or the active T3, when the terminal tyrosine is deiodinated. But at the same time, another isoenzyme which can deiodinate this internal or interior tyrosine molecule. So that gives what is called R-triiodothyronine, or reverse T3, which is an inactive T3. So this is the control and feedback regulation of synthesis of T3 and T4. Here, TRH, thyrotropin releasing hormone from hypothalamus, has an effect on pituitary gland, where thyroid stimulating hormone is produced, which reaches thyroid gland and induce the synthesis and release of T3 and T4. Now, once when T3 and T4 are released, And if the normal limit is crossed, it can have a feedback control of synthesis of itself by either acting on pituitary gland or acting on hypothalamus. The thyroid hormones are transported through plasma in bound form, bound to protein, it is an inactive form, as well as in the free active form. So when it is bound to protein, it is bound to thyroid binding globulin or to transthyretin or to albumin. And it is bound, it is called PBI or protein bound iodine, which is the concentration of which in the plasma is 10 microgram per deciliter, out of which 8 is T4 and 2 is T3. So this is a percentage transport, 70% of PBI, is thyroid binding globulin, and 10 to 15% transthyretin, and the rest is albumin. But the fraction which is made available for metabolic processes in cells is the free T3 or the unbound T4, and the free T3 and free T4, the unbound T3 and T4, which actually forms a small percentage of the total thyroid hormone that is transported in plasma. So this is the free T3, free T4, which is the active form of thyroid hormones in plasma. Now, thyroid hormones are not proteins, they are amino acid derivatives, and so there is no second messengers involved in cytoplasm. They can cross the cell membrane and enter the nucleus. They attach to specific nuclear receptors, and the receptor hormone complex binds to the thyroid response elements on DNA, and it can transcribe the required genes to be transcribed. The metabolic effects of thyroid hormones include calorigenic effect, which is thermogenesis, that means enhancing the temperature of the body, and this is being achieved by uncoupling of oxidative phosphorylation. Now, increase in cellular metabolism is another effect of thyroid hormones. That is, BMR is increased, and that's why during hyperthyroidism, body loses weight, and during hypothyroidism, body weight is increased. Now, T4 stimulates RNA synthesis, thereby increases protein synthesis, whereas T3 is having a sort of a reverse effect here. And regarding carbohydrate metabolism, thyroid hormones act like insulin in that glucose absorption and utilization are increased and stimulates glycolysis, whereas the hormones enhance gluconeogenesis and glycogenolysis also. So that's why during hyperthyroidism there can be a futile cycling of glucose. But in lipid metabolism, thyroid hormones act like epinephrine and glucagon, in that fatty acid oxidation is increased, cholesterol degradation is increased, and during hyperthyroidism, cholesterol degradation is further increased so that low cholesterol level will be there in blood, and during hypothyroidism, the blood cholesterol level will be high because there will be lowered cholesterol degradation during hypothyroidism. Disorders of thyroid hormones. Hyperthyroidism or Graves' disease. This is a high T3 and T4 and very low TSH. And this is iodine deficiency goiter, when intake of iodine by a person is low, or uptake of iodine from blood to thyroid gland is low. So here T3 and T4 are very low, but TSH is very high. TSH actually tries to induce thyroid gland to uptake iodine. So that's how the gland is enlarged with the associated problems like growth retardation and mental dysfunction. Now, hypothyroidism is when T3 and T4 are low and TSH is high, and this is mostly due to Hashimoto's thyroiditis. That is an autoimmune disease when the thyroid gland cells are attacked immunologically. And this can cause lethargy, sluggishness, called intolerance, weight gain, and associated hypercholesterolemia also.